Well, thank you very much, Takashi, for that very kind introduction. And thank you, University of Hiroshima, for this invitation to visit Japan yet again. I'm always pleased to come to Japan. And you put beautiful weather on for me on this particular trip. I'm going to talk to you today about work in my laboratory that did lead to that Nobel Prize in 2001. And I will give some historical background that actually begins at the foundation of the Royal Society in 1660. And I shall also, towards the end of my talk, give a little update on more recent discoveries and research from my lab, which are relevant to how the cell cycle or cell reproduction is controlled. But first, I just want to say a few words or introduce to you the Francis Crick Institute. We have a very exciting logo, which is what I'm going to introduce to you now. It's a dynamic logo. You can see it moves, and bang, there we have our logo. I wanted a logo that looked nothing like an academic logo, and this does not, as you can see. It is indeed the biggest biomedical research institute in Europe under a single roof. This is the architect's drawing, and this is what it looked like 2 years ago when it was opened by Queen Elizabeth, Queen of Great Britain. We have 1,200 scientists under that roof. It will expand to 1,500 over the next several years, a total staff of around 2,000. It's based in the centre of London, next to the British Library, and in the bottom right-hand corner of this photograph here, you can see the terminal of Eurostar, and we can be in Paris in 2 hours. It's been set up deliberately for that, to maintain strong links with Europe. We're not great fans of Brexit, let's put it that way. We believe very strongly in the integration of peoples and of countries, and particularly in the promotion of science. So I'm going to talk about the reproduction of cells and how this process is controlled. And my story begins, as I've already hinted, 350 years ago with the work of Robert Hooke, one of the founders of the Royal Society, which is the oldest scientific society in the world. And he, in Oxford, only 2 kilometers from where I have my house, discovered cells. He took a plant stem, took a razor, a sharp razor, and just cut a thin sliver from that plant, looked at it under a microscope similar to the one that you see up here, and in it he saw all these boxes, and you see the illustration that he drew and published in 1665 on the right-hand side of the slide there. This reminded him of a plan of a monastery, a plan of monastic cells. And so he gave the name to these boxes, cells, and that's the origin of the word. Very few people today realize the connection between biological cells and monks, but that is actually correct. This established that the cell is the basic structural unit of life. And over the next 100 to 200 years, cells was discovered in every living organism, and also single-celled organisms were discovered, bacteria, by Hooke only a few years after Hooke. In the middle of the 19th century, Rudolf Birkhoff, who was the founder of pathology, he was a German pathologist, extended the concept of the cell as the basic structural unit of life by indicating that it was also the basic physiological unit of life. And he said it in this quote, which is translated from the German in 1858, that every animal appears as a sum of vital units, that's living units, each of which bears in itself the complete characteristics of life. In other words, the cell is the simplest entity which exhibits the properties of life. So cells are not only the basic structural unit, they are also the basic physiological unit of life. And the second point that he emphasized is in that quotation from Latin, omnis cellula e cellula, all cells from cells. And that's what I'm going to be talking about today, because what that describes is that all cells arise from the division of pre-existing cells. They don't form spontaneously, as was thought by many at the time. They arise from the presence of pre-existing cells. And what we see here is a dividing human fibroblast cell just undergoing cell division. And you heard how many cells are in your body from Takashi just a few minutes ago. And since I started talking, I think you will be surprised to hear between 10 and 100 million cell divisions have occurred in each of your bodies. So this is a very important process. So why did I want to study this? Well, there are three reasons. The first is that it's the basis of all growth and also of reproduction. The second is that it links DNA replication to cells. I will explain that in a moment. And the third, it's important for medicine, particularly for cancer. So first of all, the basis of all growth and reproduction. On the right here, you see an early mammalian embryo. It's a mouse, but a human embryo would look rather similar. You can see if it's well reproduced, which it is, little spots there which represent cells. And you'll see in the diagram there how the single cell, the fertilized egg, undergoes repeated cell division to produce a mass of cells that will eventually develop into the organism. And the process of cell division is central to this, and it's the basis of all growth and reproduction. Now, I may not have yet persuaded you that cells are interesting and the division of cells are interesting. If I fail to do so, I'm going to show you this slide. This slide is a fertilizing mammalian cell. You can see the sperm and you can see the egg. And I want to remind every one of you in this audience that all of you once looked like this. Every one of you. And should this not now excite you, it's probably time to leave. This is what links us all. Here, we were all once single cells. The second reason that it is important is the linkage between DNA replication and cells. What do I mean by that? Well, what you see here is a description of the process of the cell cycle that is the basis of cell reproduction. There are two major events to every cell cycle. S phase, S at the top of this diagram, when the DNA is replicated. As Watson and Crick established in the early 1950s, the double helix is chemically replicated, and that's represented here with the two blue bars undergoing a doubling. This is a chemical reproduction. But of course, what we're referring to here is biological reproduction. And that is seen in this cell cycle, because at M phase, that's the bottom of this circle, mitosis in mitotic cells, meiosis in meiotic cells. At M phase, the replicated chromosomes, because the chromosomes are made up of the double helices of DNA, are segregated into each newly divided cell, so each cell receives a full complement of the genome. And that is true for all living organisms. So connection in the cell cycle of the chemistry of DNA replication to the cell biology of the reproduction of the cell is seen here in the cell cycle. It is, if you like, the next step after the double helix and the replication of the double helix, producing the biological consequence, which is a dividing cell. And the third reason it's interesting is that it's important for cancer. I should point out here that some of these diagrams look a little simple. They've all been hand-drawn by me, some of them 30 years ago, and I am not very good with the computer in producing nicely looking things, so I hope at the very least you'll find them charming, if a little naive. What we see here is an explanation for cancer. The blue boxes that you see there on the left would be, for example, an epithelial layer of cells in one of our tissues, that there is a defect that occurs in one of those cells, so it turns red, mutational background that results in the cell division and cell reproduction going out of control, and that leads to continued cell reproduction, and you see how the blocks of red cells get more and more obvious, and this will disturb the functioning of the tissue and the organ and eventually the organism, and is the basis essentially of all cancer. So understanding cell reproduction is also central for understanding diseases such as cancer. Now, I hope I've convinced you this is an important process, and understanding how it is controlled is important. And an issue here is how can this be investigated? And when I started working on this problem, which is now many years ago, in the early 1970s, very little was known, in fact, really nothing at all, about how cell reproduction was controlled. And after I finished my graduate studies, I decided this was a problem that I wanted to focus on. I decided that the way to approach this problem would be to use a very simple organism, a single-celled organism, and I used the fission yeast Schizosaccharomyces pombe. You can see a scanning electron micrograph there on the left. It's a very simple beast. When I started working on it, it's a fungus, of course, but when I started working on it, it was thought to be a plant. Now, 40 years later, it's thought to be an animal. Of course, it's really something in between. But the advantage of working on this organism is that it's simple. It only has 5,000 genes. We have 25,000, so it's a little easier to investigate. My lab discovered the 5,000 genes because we organized the sequencing of this organism. And I put for your interest another picture on the right, which is the first recorded description of fission yeast, which was in the 19th century, because it was discovered in the Carlsberg Brewery in Copenhagen. I actually spent several months working in the Carlsberg Brewery earlier in my life. It was discovered as a contaminant in a bad beer, because good beer is made by budding yeast, and when it gets contaminated by other microbes, bacteria, or in this case by fission yeast, the beer goes bad. So I'm afraid, ladies and gentlemen, fission yeast is mostly useless for humankind, except perhaps for the investigations that I'm about to describe to you. So this is fission yeast, and what is good about it is that it's very convenient for genetics. And this was established by my two mentors early in my life, who worked on fission yeast, Urs Leopold in Switzerland, in Bern, and Murdoch Mitchison in Edinburgh. And I worked with both of these, who were tremendously supportive early on in my life, and who established this organism as a laboratory organism. Let me describe to you the principle by which you can use genetics to investigate a complex process, such as what controls cell reproduction. And that's really shown here. The box here on the left at the top represents a cell, and the green circles that you see there, a dozen or so of the 5,000 genes that are present. Now, if you have a process like the cell reproduction that you don't understand, you can use genetics by randomly damaging all of the genes and selecting for strains with a damaged gene that influences the process that you're interested in this case, the cell cycle and cell reproduction. And that way, you can identify the set of genes that you should be interested in if you're trying to understand a process such as the control of cell reproduction. So you mutagenize the cells, and then you look at all the individual colonies that grow up and look for phenotypes, that is, characteristics in the cells which are likely to be revealing about the process that you're interested in. In the top, you'll see cells that are perfectly normal and can produce dividing cells. In the bottom part, where you see elongated cells, these are a consequence of a gene being damaged that is needed for the cell cycle. So it cannot reproduce, cannot divide, but can continue to grow, and so produces highly elongated cells. This is not rocket science. This is very simple, but it gets you into the problem. Because what that does is allow you to identify the genes that are required for this cell reproductive process. They have to be what we call conditional mutants, because obviously if they die all the time, you can't keep them alive. So this mutant phenotype can only be expressed under certain conditions, usually in high temperature, as was the case here. When I undertook these studies, my colleagues and myself, this was in the 1970s, originally identified 30 CDC genes, CDC standing for cell division cycle genes. So we identified these genes as ones that were required for cell reproduction. Now, this was only a small percentage of the total. We now know that there's around 300 essential CDC genes. I know this because we, as I've already said, sequenced fission yeast. It was the third or fourth eukaryote to be sequenced. I did it in collaboration with laboratories, which was coordinated by my laboratory together from the Sanger Institute across Europe. And we sequenced the genome and identified 5,000 genes. And then Jackie Hales in my laboratory led a South Korean consortium to delete each one of those genes one at a time. So we now have a fantastic resource where of 5,000 strains, each of which are deleted for one single gene. And this allowed us then to identify every single gene that was required for the cell cycle in an organism. It's the only time this has actually been done. And we know there's 300 essential such genes. Now, what you see here is a cartoon that explains what I've just been talking about. In the middle there, what you see is a normal cell cycle. The cell mass or cell size is versus time. As cells grow, the nucleus, that's that black blob, divides, the cell divides, and it goes back to its original size. If it cannot undergo cell reproduction, it produces these elongated CDC cells with a cell cycle block. Now, this approach defines genes that are necessary for cell reproduction. But you may recall that I was interested in what controls the cell cycle, not simply what is needed for the cell cycle, but what controls it. Now, this needs a discussion of what do we mean by control? And it can have several meanings. It can mean how you start a process, and it can mean what determines the rate at which a process occurs. And both of these definitions I've used in my work. The first concerns what determines the speed or the rate at which a cell can reproduce itself. And the logic behind this approach is also shown on this slide. Because if you get a mutation in a gene that is rate limiting for cell cycle progression, then one possible outcome of that is that the cell will complete the reproductive cycle more rapidly than normal. Should it complete the cell cycle more rapidly than normal, it will divide before it has grown to the normal extent. This means that it will divide at a small size. So to find cells which are altered in genes, mutants that are altered in genes that are rate limiting for cell cycle progression, you look for cells which divide at a small size. And that is shown here on I should have just shown you this, actually. This is what the elongated cells look like in the CDC mutants. If you look here, you will see under A, these are normal cells. Do you see that bright white line? That's a dividing cell. And if they can't divide, you see in B and C, they just become highly elongated. This was a graduate student working with me at the time, Kim Naismith, who's now Professor of Biochemistry in Oxford. This is the other type of mutant that I was just describing to you. A is wild-type cells under a different form of microscopy, and B shows the mutant, which I called a wee mutant. For those of you who have ever visited Scotland, you may know that the word wee is used as slang for small. So I called these mutants wee mutants. I thought it was quite funny at the time. I'm not so sure it's so funny 45 years later, but there you are. This gene, therefore, was Wee1 that was mutated. And one of my colleagues there is Peter Fantes. Now, I'd like to tell you that I thought about this problem in the way that I just described to you here. It isn't true, however. What I was doing was looking for mutants which were elongated under the microscope, and I saw a mutant that looked like B here. And looking down the microscope, and I remember it still, it was like a light bulb waking in my brain. I looked at this, and within about a minute, I realized what it meant, but I had never thought of it beforehand. I mention this only because sometimes in biology, at least, it's nature that leads you somewhere if you keep a track and observe what is happening. And it's not always just the human mind constructing things, it's also looking to see where nature is taking you. Having, however, seen this, having been led here by nature to discover these wee mutants, I then decided that these were going to be important for understanding control. And if they were important for understanding control, then I needed to collect wee mutants. This was a very tough task. There is no way to actually select for them. You can only visually scan and look for them under the microscope. I could tolerate about 3 or 4 days a week doing this, and during a week I might get 1 or 2 such mutants. And so I gave myself the task of isolating 50 such mutants, which took me around 1 year or thereabouts. It was very tedious, ladies and gentlemen, I can tell you, doing this project. And what I hoped to do was to discover all the genes that could be mutated this way. But every time I got a new wee mutant, I crossed it to my original wee mutant and found it was the same gene. Every single one, up to either the 47th or the 48th mutant. So this was getting a little depressing. I found a mutant, number 47, number 48, on a wet Friday afternoon in Edinburgh in November. And I was feeling a little depressed. You're going to hear about depression a little bit later today, by the way. I was feeling a little depressed. And it was covered, the plate where this mutant was, it was an agar jelly plate, with a fungus. And those of you who have ever tried to do experiments of this sort will know that is awful, because the fungus spreads so rapidly that it contaminates everything in the plate. So I threw it away in the rubbish, and I went home to have my tea. Then I felt guilty about it. I kept thinking of this plate in the rubbish with this mutant, and I cycled back and took it out of the rubbish and started to re-isolate it. It took me about 10 days to re-isolate and purify this one strain. But you know what I'm going to tell you next. This was the only mutant out of the 50 that was not Wee1, and it was a gene that I called Wee2, so I got a second gene. Now, the fact that I got 49 out of 50 in Wee1 suggested that this was a relatively common mutation, and therefore it was likely that Wee1 encoded a protein that was acting as an inhibitor of cell cycle progression. So if we mutated it and took it away, cells would be advanced through the cell cycle prematurely because we'd removed an inhibitor. You understand the logic. So I did further tests, which I won't explain to you. This is what I looked like whilst I was doing these experiments, by the way. Don't get frightened by it. Everybody looked like this in the 1970s, of course. But from this, I concluded that We1 was an inhibitor, a negative element. We2, on the other hand, was very rare. So I wondered whether, in fact, this was required positively so that I was mutating it, a rare mutation, so it was more active than normal. And therefore, if it was mutated so it wasn't active, the cells wouldn't be able to divide at all. And I was encouraged to think that because the mutation was dominant. But what this meant was, what was possible that it could mean, was that the 30 CDC genes that I'd already identified, amongst them might be the equivalent of We2, because it's, if We2 lost its function, it wouldn't be able to divide. So I crossed We2 to all the 30 CDC genes, and We2 was linked to CDC2. It was the same gene. So this meant that I had one gene, CDC2, which could be mutated to lose function, and therefore cells couldn't proceed through the cycle, or it could be mutated to have a gain of function, in which case it went more rapidly through the cell cycle. I was excited by this, and I did analysis of the mutant and discovered that it functioned late in the cycle at the onset of mitosis, and so the major rate-limiting step, as revealed by this mutant, for cell cycle progression, was occurring at that G2 to mitotic transition. But it turned out to have a more complicated phenotype. This I've drawn that cell cycle again. You'll see it's now called P34CDC2. I'll explain why in a moment. But more analysis of the CDC2 mutant showed that actually CDC2 was required twice in the cycle. It was required in G1 to initiate S phase, to initiate DNA replication. And this was, if you like, the start of the cell cycle. Remember I said that you can think of control as being rate limiting or starting a process. And my colleague Lee Hartwell had discovered this start process in the budding yeast. He also won the Nobel Prize. And CDC2 was involved in this start process in G1. And why I therefore became excited is that one single gene, CDC2, seemed to play a key role in controlling the start of the cell cycle and the transition into S phase, and the end of the cell cycle, the transition into mitosis. Now, up until now, my analysis, because it was carried out in the 1970s until early 1980, is completely abstract. I haven't told you anything about what this gene does, because I had no idea what the gene did. To do that, you have to be able to clone the gene and to work out the function. But this was right at the very beginning of cloning. It was in the late 70s when I started this, when cloning had only been around for 1 or 2 years. So I turned my lab into a molecular genetics lab to introduce molecular genetics into fission yeast that hadn't been done previously. And that meant I had to find ways of introducing DNA into cells, of deleting genes, of modifying genes, and producing gene libraries. In fact, what we now call gene editing with CRISPR-Cas, you could do in yeast in the 1980s by a different method, which is one reason why yeast was so powerful at that time. So having developed those techniques, this allowed us to clone the gene. And I want to explain that again with one of my hand-drawn slides that I made in around 1980. And what we did is we made a gene library. We took fission yeast DNA, we cut it up into small pieces, we cloned them into other DNA plasmid molecules, which are circular, and so we would have a library of the 5,000 genes. We could then sprinkle them onto fission yeast, and they'd be taken up with the conditions that we used and introduced into the cells. If we sprinkled them onto a CDC2 mutant, which die because they can't complete the cell cycle, then we can select for a gene that has the CDC2 gene because it rescues the defect on the chromosome. And you see that on the right-hand panel there. A plasmid, which contains the CDC2 gene, makes the CDC2 protein, which is defective in the genome. These cells can now grow and divide. We form a colony on a plate. We can isolate that plasmid back out of those cells, and we have the CDC2 gene. That we did, and it worked. We then had to sequence the gene to find out what it did. This is now the 1980s. I just want to tell you how long it took. It took 15 months to sequence 2,000 nucleotides. I got advice from Fred Sanger on how to do it, who had invented DNA sequencing and got 2 Nobel Prizes for protein sequencing and DNA sequencing. It took 15 months. We can now do it in about 15 seconds. It's just extraordinary advances since that time. We sequenced the gene, but there were only 100 gene sequences in all the world at that time. And half of these were actually cytochrome C, which had been protein sequenced, and so weren't really useful at all. CDC2 only found any identity with one gene, and that was SARC-P60. This is an oncogene in the SARC virus, which was thought to encode a protein kinase. And so we decided to see whether CDC2 was a protein kinase. This required to do experiments, biochemical experiments, which had not been done very much before. What we did is we expressed the CDC2 gene in bacteria, we purified the protein, we put the protein into rabbits, we made antibodies, we used those antibodies to immune precipitate the CDC2 protein out of fission yeast cells. We then bought a lot of different proteins from the Sigma catalog, for those of you who are biochemists, and we added this immune precipitate containing CDC2 to maybe 50 different substrates, and we found that radioactive p32 phosphate was incorporated into 1 or 2 of the proteins, and the most effective at that time was a milk protein called casein. So we had an assay. And we could show, using that assay, that the CDC2 protein kinase was responsible for that activity because we used a temperature-sensitive CDC2 mutant and showed that in vitro, the kinase activity we were measuring was lost if we shifted the extract to high temperature. So we knew exactly what we were doing there. For those of you not familiar with protein kinases, I've put this cartoon up here. You see under B, the protein kinase has a red blob, which is the phosphate, and that can be attached to a variety of substrates, S1 to S5. If you introduce such a negative molecule into a protein, negatively charged molecule, it can change the structure of the protein, as I tried to show on 8. And if you change the structure of the protein, you change the characteristics of that protein. And what this means is that protein kinases can cause an alteration on many different substrates. So that it acts as a very powerful molecular switch. And that's how protein kinases actually work. So at this point, we concluded that in fission yeast, the CDC2, the major cell cycle control was due to a CDC2 protein kinase, and the CDC2 protein kinase was required to begin the cell cycle and to end the cell cycle. The problem with this was that it was yeast. And I'm realistic with you, most people in the world are not that interested in yeast. They tend to be interested in human beings or mammals at least, or maybe chickens or at least some other organism, but not so much yeast. So the question was, is there a CDC2 gene equivalent in humans? So that was the task that I next set in the laboratory. Now the usual way, and this was in the mid-1980s, the usual way of identifying across species, a protein or a gene, is to look for structural similarity, either using antibodies to look for a similar protein or to look for DNA sequence. And for about 1 or 2 years, a postdoc in my lab, and I'll show you a picture of her in a moment, used these conventional techniques. We isolated 1 or 2 protein kinases, but by this time we were aware that there were hundreds of protein kinases in cells, and we had no idea if it was the right one or not. Do remember that yeast and human beings diverged from each other 1.5 billion years ago. That's 1,500 million years. And we were demanding that these proteins would still be similar. So this was a very unlikely case. So we decided to try and clone it not by structure, but by function. So we repeated this experiment. Which I don't need to explain to you because I've explained it already. But instead of using a yeast library, we used a human cDNA library. Now, the human cDNA library was made by Hiroto Okiyama, who some of you may know. He worked at Tokyo University, and Paul Berg. And they made the library. I saw it was published, and they gave it to me only a month after they published it, just freely. And I've always been grateful for that generosity. I used that library, transformed it into fission yeast, and it found this approach, the human CDC2 gene. Because in fact, despite 1,500 million years of evolutionary divergence, the human protein still worked in fission yeast and could still control the cell cycle in fission yeast. And this is Melanie Lee, who did that work. And what you see here is the sequence that was that we got, the amino acid sequence, which showed 61% identity with between the human and the yeast genes, despite that enormous evolutionary divergence. I took a picture of the very first cells being kept alive by the human CDC2 gene, a very crude photograph, just pushed a camera onto the eye, and this is it. This is when we thought it was likely that this was conserved. Because if it's conserved between yeast and humans, it's conserved in everything. And we actually showed it was in flies and plants and so on as well. Now, I won't go into the evidence for this, but CDC2 is a protein kinase, and it's complex with another molecule called cyclin. So that these form a family of cyclin-dependent kinases. Cyclins were discovered by my friend and colleague at the time, a colleague in the Imperial Cancer Research Fund, Tim Hunt, who is now in Okinawa, in OIST, in the new institute in Okinawa. And he's also a visiting professor in the Crick Institute as well. And it is this cyclin-dependent kinase, the kinase and the cyclin, which is responsible for the start of the cycle and the end of the cycle. It also has a third function that I just want to introduce to you. And that third function is to block DNA replication. And this is shown in this experiment here that we did in the 1990s, where we completely destroyed the CDK complex. And if we did that, then the cells would simply get locked into continuous DNA replication. And can you see those little round bright circles? They are wild-type cells. And you see the big cells with this is stained for DAPI, which stains DNA, which have undergone repeated rounds of DNA replication, and the DNA content has now gone up 50 to 100-fold, because they just go repeatedly round DNA replication. There's a FACS analysis there under C. So what this told us was that the cyclin-dependent kinases start the cell cycle and bring about S phase. If they have a bit more activity, they block S phase, and that's the reason why you only have one S phase per cell cycle, and then you bring about mitosis. And this seemed to involve a variety of CDKs. This is fission yeast, and there were 4 cyclins, PUC1, SIG1, SIG2, CDC13, thought to act at different stages of the cycle, 1 CDC2 CDK. In more complex organisms such as our cells, there's a family of cell cycle CDKs, more cyclins, more protein kinases. So it was getting very complex. And the regulation was very complex. Here's a slide which shows that cycling CDKs are driven by a whole collection of different types of regulation. And I was getting a bit confused by this. There was so much regulation, it was difficult to work out what principles were underlying what was going on. This is all post the Nobel Prize, I should say, so I've moved on from the Nobel Prize work. So we decided in the lab to see if we could simplify it. And the first step we did was to examine the fact that there are two components, the kinase and the cyclin, and they have to find each other and form a complex. So what we reasoned was, if we made a chimeric molecule, a single gene, that would simplify the system and remove some of the controls. So we made a chimeric protein of CDC13 cyclin fused to CDC2. Introduced that into fission yeast. Here is that experiment. You can see wild-type cells dividing normally. You can see CDC13L, that's a linker between CDC13 and the CDC2 protein in the middle one, and we deleted the CDC2 gene and the CDC13 gene in the genome. So the only kinase here is CDC2, and you can see these cells are perfectly normal. Then what was done here by Damien, a postdoc in my lab, is he deleted the other three cyclins required for the mitotic cycle, SIG1, SIG2, and PUC1. So we deleted all four cyclins and the native CDC2 and replaced it with a single chimeric CDC2, CDC13. Bit complicated, but what that means is that this whole cell cycle is now being driven by just one cyclin-dependent kinase. It doesn't require all the CDKs. There's evidence, of course, in mammalian cells that these are very plastic and that there can be substitutions. And we're taking this to an extreme here. This is for the mitotic cell cycle. If we look here at the meiotic cell cycle, and you don't really need to look at it, you just need to look at me, actually, because I'm not going to explain it, there are 6 CDKs required for the meiotic cell cycle. So we then did the same experiment here. We deleted all 6 cyclins required for meiosis and showed that they could be replaced by that single CDK. So in fission yeast, we can drive all of the mitotic and meiotic cell cycle with only CDC2 and CDC13, as long as it's in that chimeric protein. So why are there so many CDKs? Well, I don't know that for certain, but I think that the primitive eukaryote had a single CDK. This underwent gene duplication, and more specialized functions arose with different regulation and so on, and that this led to addiction to a range of different CDKs, but under the right expression conditions, which we have with our chimeric protein, we can substitute all of those by a single CDK. And I think that's going to be very likely the case for mammalian cells as well, except when you have to make tissues and organs in an organism, it will be more complex. But for making cells, I think a single CDK is functional. And this has led us to this hypothesis here, which is, in fact, a very simple hypothesis. And the idea was that what was the principle underlying the cell cycle and its control? We'd shown that cyclin-dependent kinase was critical, and what this idea suggests is that what happens is that CDK activity increases continuously through the cell cycle, and that's shown by the depth of the red color. It starts off being very low. You then get a small amount of activity, the sort of light-colored pink, and you get S phase. You then get more activity, and that suppresses S phase, so there's only one S phase per cell cycle. Then you get more activity, and that brings you mitosis. And then you kill all the activity by degrading all the cyclin, and you start the whole thing again. In other words, the principle is a very simple one. Continuing increase of cyclin-dependent kinase, differential sensitivity of substrates, required a S-phase G2 and mitosis to that CDK activity, and that gives you the temporal order of cell cycle progression. And I'm going to finish with a couple of experiments that, that support that idea using phosphoproteomics. And what we did was to do a phosphoproteomic investigation through the cell cycle to ask what proteins were phosphorylated at different stages of the cell cycle. And we did it in a very careful way. I won't explain why it was so careful, but we made sure that we were only looking at CDK substrates, absolutely CDK substrates. And to our surprise, we got this pattern. You see that we have the cell cycle at the bottom, and you can see, I hope, blue compared with purple. Blue are S-phase, G1 and S-phase CDK substrates. And you can see as cells proceed from G1 into S-phase, there is an increase in their phosphorylation. And that is responsible for the start of the cell cycle. To our surprise, that didn't go down. It continued to increase through the rest of the cycle. But because now increasing CDK activity inhibited S-phase, then you didn't get any more DNA replication taking place. In contrast, phosphorylation of M-phase substrates didn't occur at all during this phase of the cycle and only became apparent at the end of G2 at the onset of mitosis. And you can see that with the purple substrates, which are the mitotic CDK substrates. And then all of them become dephosphorylated when the cyclin is destroyed at the end of the cycle, because the turnover of these phosphosites is very, very rapid. For those of you who are familiar with this field, the half-life is only about 2 minutes. So it's a very dynamic system. So if you kill the cyclin-dependent kinase activity, then they will become rapidly dephosphorylated. So that's the principle. We then did a final experiment, which I'm going to explain to you, which was to measure in vivo protein kinase activity. The normal way that we measure protein kinase activity is to break cells open, make an extract, add a substrate, and add radioactive phosphate, and look for incorporation into the substrates. But that doesn't tell you what the in vivo activity is. What it tells you is what the in vitro activity is when you extract the enzyme. So we developed an in vivo assay, which hasn't been used before. What we did was we added an inhibitor to the CDK. Because the half-life is only 2 minutes, within 10 minutes or so, all the substrates are dephosphorylated. Then we took the inhibitor out and let the kinase activity return to cells, and then we used the mass spectrometer and phosphoproteomics to measure the rate of phosphorylation of hundreds of substrates simultaneously. And you can see there that if we did that from G1 cells, this is the top left, or S-phase cells, G2 cells, or mitotic cells, that there was an increase in that in vivo kinase activity from very low in G1 to very high in mitosis. And you can see we've done an instantaneous rate of CDK restoration at the bottom to measure that activity. So this provides a method to measure in vivo protein kinase activity and shows that in vivo, the CDK activity increases as you go through the cycle. And on the bottom, on the top right in black, you can see that what we looked at was the rate of phosphorylation in these experiments in substrates that are phosphorylated late at mitosis, in the middle in G2, or early at G1S. And you can see substrates that are early are much more readily phosphorylated than substrates that are used late. So the principle for temporal order in the cell cycle, why you have S-phase before mitosis, is that CDK activity rises and that there is a differential sensitivity of the different substrates required at these different steps. So, just to return to this slide, when there is a small amount of CDK activity present, you get S-phase. When you have more CDK activity, you phosphorylate substrates that inhibit S-phase. And when you go higher, you phosphorylate substrates that undergo mitosis. This could not be a simpler system. It's become more complicated with redundancies and the human mind, to be quite honest, but this system really couldn't be simpler. If we destroy activity in G2 and drop it down and let it rise again, then these cells will re-replicate. So there is no arrow of time in the cell cycle. You set where you are in the cell cycle by the level of cyclin-dependent kinase activity. Ladies and gentlemen, I apologize. There will be some of you in the audience which will have found the last 10 minutes to 15 minutes a bit difficult, but I just wanted to bring this up to date. So my conclusions. Cyclin-dependent kinases control the eukaryotic cell cycle. There is a simple principle. Increasing activity drives, first of all, the onset of S phase, then inhibits, and then S phase, and then drives the onset of mitosis. You have multiple CDKs, which have specialized functions, but under the right conditions, you only need one cyclin-dependent kinase activity. So the principle is quantitative level of CDK activity rather than qualitative differences between different CDKs, which is what you will read in most papers and textbooks. And finally, the control is the same in all eukaryotic cells. So once this was established, probably 1.5 billion years ago, it's remained the same, because to change it after that would just kill all the cells. So you get locked in to this way of functioning. Now, I think when we discovered that it was the same in yeast and humans, there was some surprise, because we like to think perhaps we're more important than yeast. But in fact, this idea was anticipated. And I'm going to end my talk with a quotation from Theodor Schwann in 1839, as you can see here. He said, and I should just quote it, it was in German, but this is an English translation. "We have seen that all organisms are composed of essentially like parts, namely of cells." That's the cell theory, okay? "That these cells are formed and grow in accordance with essentially the same laws. Hence, that these processes must everywhere result from the operation of the same forces." Now, this is saying exactly the same thing as I have just told you, but it was said in 1839. It just took us 150 years to turn this proposal, philosophical proposal, into an experimental reality. But it was anticipated by Theodor Schwann. Well, thank you for listening, ladies and gentlemen. I now will finish with destruction of my logo. Bang. Thank you very much.